Hello everyone, this is K 2023 Life Sciences, Section P Biochemistry, question number 40. Match the cell types listed in Group 1 with associated processes listed in Group 2. So Group 2 consists of different types of cells like NK cells, B cells, mast cells and neutrophils. And Group 2 consists of their function. Here first is antibody production. First cells to be recruited at the site of infection. 3. Antibody dependent cell mediated cytotoxicity. 4th, histamine production. And these are the different options after matching group 1 and group 2. This is the NPTEL online course reference wherein you will find the basics of immunology. So first we'll start by understanding these different types of cells and their functions in our cell. First we'll start with B cells. So B cells originate from a stem cells. So this stem cells undergoes gene rearrangement to mature into B cells. So 1, 2, 3, 4 are mature B cells which are committed antigenically to form B cells. So when they are committed to become a mature B cells, they specific isotopes, IgG chains which are found on the surface of this B cells, which are the signature molecules represented on the B cell. Okay, these cell markers will always be present on a B cell. So now these cells are committed to form B cells in bone marrow. And from the bone marrow they are further transferred to lymphoid organs wherein they further mature and meet the antigens. Also in bone marrow, before going to the lymphoid organs, they undergo self and non-self recognition, wherein so the B cells which recognize the self antigens are destroyed in order to avoid autoimmune diseases and autoimmune responses inside the body, and the B cells which do not recognize the self antigens are further matured and then they go to lymphoid organs. So in the lymphoid organs, the mature B cell recognizes the antigen in peripheral lymphoid tissue. So among the population of B cells, like 1, 2, 3, 4 shown here, only few B cells will recognize the antigen. So only those B cells which are able to recognize the antigen are further proliferated and differentiated into different populations of B cells, and those which do not recognize any antigens in the peripheral lymphoid tissue are again destroyed, right? So only this second B cell recognizes the antigen, whereas 1, 3, 4 do not, and hence 1, 3, 4 are destroyed in the peripheral lymphoid tissue, and second B cell undergoes antigen dependent proliferation and differentiation. So after proliferation of these B cells into many clones of these second B cells, they undergo differentiation to form plasma cells and memory cells. Okay, these are the two important cells which B cells differentiate into. So first we'll talk about these plasma cells. Okay, so the plasma cells are special types of cells which are able to produce antibodies, right? So the antigen which was recognized in the peripheral lymphoid tissue, this antigen recognition these antigens are further processed inside the plasma cells, and they release antibodies specific to that antigen. Okay, and these antibodies are released at a very high titer in the blood to fight against the antigen which was lymph tissue. Okay, so plasma cell functions to produce large number of antibody titers inside the body, whereas B cell further divides into memory cell. Okay, this memory cell is also equally important So whenever the antigen is encountered, the B cell will proliferate into memory cell, that is, it will have the memory of that antigen. So on the second encounter of this antigen, there will be a large dose of antibody production from the plasma cell. So whenever the antigen is encountered by the B cell, it differentiate into memory cell of that antigen, and it differentiates into plasma cells, which produces a huge titer of antibodies to kill the antigen or to destroy the antigen which is present in the blood. So this memory cell has a high lifespan as compared to that of plasma cell. Plasma cell lifespan is a very short lifespan. Its only job is to produce antibody and then the plasma cell is destroyed. Whereas the memory cell can live up to years and years inside the body. Example of this memory cell is when we have common cold by rhinovirus. So after the change in every season, like 3 to 4 months, we get common cold. It is observed that due to seasonal changes we have viral cold infection, and the memory cell lifespan is about 3 to 4 months, and hence once you get a common cold viral infection, it is very rare that you'll get the once again you'll get the same viral infection in those 3 to 4 months. So you have the memory for 3 to 4 months, and hence your body will fight to that virus which causes the common cold. Also, we know that the chicken pox viral infection which we get as kids, the memory stays for the lifetime. Why? Because the memory cells have a lifetime. Memory cell has a lifespan of n number of years. Like, very unlikely that a person will get chicken pox twice in his lifetime. Okay, so the memory cell has different lifespan it totally depends on the type of antigen, whereas plasma cells has very short lifespan. Okay, so the main function of B cell is to produce memory inside the body to recognize the antigen and fight against it on its second time encounter. And during the first encounter, the B cell produce large titer of antibodies. So the next cells are mast cells, and these mast cells are commonly involved in various allergy related responses. So whenever a B cell, as we seen earlier, recognizes an antigen, it produces a memory cell and plasma cell. And with plasma cell, it releases large number of different types of antibodies. So whenever an allergen is encountered by a B cell, it produces large number of IgE molecule. That is the one of the property of different type of allergens. These allergens mostly induce production of IgE antibodies from B cells. Okay, when they are ingested by B cells, these allergens, B cells will produce IgE molecules, and these IgE molecules released into the blood are further attached to the Fc receptor, which are present on the mast cells. So the mast cells has a specific Fc region which recognizes these IgE molecules, and the Fc region of IgE molecule binds to these Fc receptor present on the mast cells. Okay, so as you can see, the mast cell has a cytoplasm. It contains various granules, and these granules are filled with substances like histamine and various cytokine, which further elicits allergic reactions. Okay, so whenever the allergen is attached to this IgE molecule, so when B cell releases IgE molecules specific to the allergen, these IgE molecules attach to these mast cells along with the allergen as shown here, and they releases large amount of and the granules are released into the cytoplasm. There the degranulation process occurs, and these granules which are filled with histamine and other substances are released into the cytoplasm, which further induces allergic reactions. Okay, so next cells are natural killer cells. Natural killer cells also originate from common lymphoid progenitor cells, where T cells and B cells are T cells and B cells are differentiated. Natural killer cells, as the name suggests, they kill virus infected cells and tumors. So natural killer cells expresses these receptors KIR, CD94 and NKG2. Okay, so these receptors are present on the NK cells, and along with this we have also AR receptor, which is activator receptor. AR is activator receptor, and this KIR is inhibitor receptor. Okay, killing inhibitory receptor. So the green one is will have an inhibitory effect, and the activating effect is the AR receptor. I'll explain you what are the roles of these two receptors. So the AR activatory receptor will bind to a AR ligand on a normal cell. So this is a normal healthy cell. This one, it is not infected by any virus or it is not tumorigenic. Okay, it and normal healthy cell, they have a AR ligand. So the AR, when this AR ligand binds to the AR receptor here, when this binding occurs, it gives a positive signal. Okay, so activatory receptor gives a positive signal to NK cell, and since they are natural killer cells, so their positive signal will be to kill the cells, right? So the positive signal for natural killer cell is to kill, and that's why and thus the cell will undergo death unless there will be a inhibitory signal by KIR or CD94 or NKG2. Okay, so this inhibitory receptor on NK cell will inhibit. Okay, it will have negative role on killing, and thus it will inhibit the killing. Okay, so class 1 MHC molecules, this is very important. Class 1 MHC molecules are expressed on all nucleated cells, right? It is expressed on all nucleated cells. Any cells which has nucleus, they will express class 1 MHC molecules. This is a fact that you should know, that all the nucleated cells inside the body always expresses class 1 MHC molecule. So in normal healthy cell, they will have a healthy expression of this class 1 MHC molecule. And the inhibitory receptor KIR or CD94, this receptor has affinity for the class 1 MHC molecule. Okay, and thus when a normal healthy cell binds to NK killer cells by activatory receptor, so we'll have the first killing signal to kill, but will be bind to the class 1 MHC receptor present on a normal cell, which will give a inhibitory signal to not kill the cell. Since it is a healthy cell, it is healthily expressing class 1 MHC molecule, the NK cells will tell themselves that no, this is a healthy cell and we are not supposed to kill the cell. So that's why this this type of mechanism is two receptor model, okay, for NK cell, for natural killing cell, for natural killer cells to kill the cells, okay. So this is a two receptor model in which they have a activatory signal from AR receptor and they have a inhibitory signal from KIR receptor, okay, very simple. So when the normal cell has a healthy expression of MHC class 1 molecules, there will be no killing, okay, and the cells will be healthy. The cells will proliferate and divide, okay. And what happens in case of virus infected cells or tumor cells? In case of virus infected cells, the class 1 MHC expression levels, the expression levels of class 1 MHC goes down. So now a normal, the virus infected cells will have very less MHC, class 1 MHC receptors on them, or they will completely will not have any class 1 MHC receptors, and thus when natural killer cells will recognize through activator receptor, they'll first recognize and activate a killing signal. So there will be a killing signal which is activated, and this killing signal will not be inhibited. The killing signal is activated and this killing signal will not be inhibited since there are no MHC molecules here to bind to the inhibitory receptor to give a inhibitory as we observed in the normal cells. Okay, so this is how natural killer cells kill virus infected cells or tumor cells. Okay, and once there is a positive signal to the NK cells that yes, we have to kill these cells because these cells are not expressing MHC class 1 molecules, so these cells are not healthy. So what they do, they releases various perforins and granzymes. They releases perforins, granzymes and cytokines like TNF, interferon gamma, okay. They releases this and this will act on this neighboring virally infected cells, okay. And the perforins will cause pores inside the cells and it will cause cell death. Similarly, granzymes will also lead to cell death. It will it has enzymes which lyse the cell wall and it will ultimately lead to cell death. TNF ligand also has a cytotoxic role. So all these together will act on that neighboring cell, right, and it will kill this cell. So this is how the NK cells kill. So now this interaction is known as antibody dependent cell cytotoxicity, also known as ADCC, right. So there is receptor ligand, there is antibody antigen, the ligand and antibody interaction here, and thus this process is known as antibody dependent cell cytotoxicity. So natural killer cells originate from a myeloid progenitor by hematopoiesis process. So the cells which we studied before, natural killer cells, B cells, these all cells originated from a lymphoid progenitor, whereas neutrophils originate from a myeloid progenitor. So neutrophils are granulated, right? They have so they are granulated and other granulated, these cells are granulated, that is they have granules inside them, And apart from neutrophil cells, other granulated cells are eosinophils and basophils. So skin is a protective layer, and the bacteria cannot enter into the bloodstream unless and until there is some tissue damage or cut. And once there is a damage, the bacteria, as you can see, has entered into the system. So what happens when this bacteria enters in the bacteria enters in the body, that is because of tissue damage, and also because of tissue damage, it causes releases of various vasoactive and chemotactic factors. Okay, and these chemotactic factors and vasoactive molecules increases the local blood flow. Okay, so in the site of infection or the site of cut, we require various cells to be recruited, and in order to do that, these vasoactive molecules increases the blood flow. They increase the blood flow, so many cells will flow to the site of infection, and the immune cells will flow to the site of infection to kill off these bacteria. Okay, so the blood flow is increased. Also, the blood vessel, the capillary permeability is also increased, so that the cells will squeeze off from these capillaries, right? So there is vasodilation, right? The capillaries dilate so that there is increase in blood flow. Okay, and there is the capillaries, the blood capillaries increases the permeability so that the cells can squeeze through. So this is the first step, is vasodilation and vascular permeability. Vasodilation and vascular permeability. Okay, so now all the cells are now alerted that okay, we have a problem, there is tissue damage and there is a bacteria which has entered into the body. So there is influx of various neutrophils. As you can see, these neutrophils are the first line of defense. Whenever a bacteria enters into the system, neutrophils rush to take action, and these neutrophils, first they'll flow and then they'll migrate towards the site of infection, right? And then they'll squeeze through, right? They'll squeeze through the capillaries. As the permeability is increased, they'll squeeze through the capillaries, and these neutrophils are responsible for phagocytosis. Everybody is familiar with phagocytosis, right? So in phagocytosis, what happens? There is first this is the bacteria, there is the membrane gets surrounded to the bacteria, right? The bacteria is completely surrounded by the membrane, and it engulfs this bacteria, and then it fuses it forms phagosome, and this phagosome then fuses with lysosome, which has various enzymes to kill the bacteria, right? This is a very common phagocytosis process. So this neutrophil cells are responsible for phagocytosis. Okay, so what happens when there is a signal for these neutrophil cells by tissue damage and bacteria? So bacteria also releases few signals like endotoxins, which trigger neutrophils to rush towards them. So there is first rolling on the capillaries. This is the endothelial capillary. First there is a rolling. Okay, the next step is activation, and then it adheres to this endothelium cells on the capillary. So these are the various receptors which are present on endothelium: E-selectin and IG superfamily CAM receptors. So CAM integrin forms adhesions. These helps in adhesion. Okay. And E-selectin binds to a CAM mucin. So wherever there is a selectin molecule, it will always bind to mucin like CAM, which are present on the neutrophils. And there is also chemokine, which is a chemoattractant. So whenever when there is damage, the tissue will release chemokines, and these chemokines will be attracted by a chemoattractant receptor present on a neutrophil, so that they can rush. And then there are cell adhesion through these receptor ligand interaction, and then finally the neutrophils are squeezed through the endothelium layer of the capillaries, and then they eat the bacteria. They destroy the bacteria by process of phagocytosis. Okay. So neutrophils are the first line of defense, where whenever there is bacterial encounter, these cells will come and they'll kill off the bacteria. So coming back to our questions, first we'll start with neutrophils, which we discussed now recently. And so neutrophils are the first cells to be recruited at the site of infection, right? So this is S is 2. And mast cells, always remember, mast cells release. So mast cells release histamines, and it leads to allergic response, right? Allergic response due to any allergen like pollen, any allergen which is responsible. Mast cells with the help of IgE. IgE is the immunoglobulin which is responsible for allergies, right? And this IgE is released by B cells, right? B cells will always release antibodies, and this antibody IgG is released in response to the allergen from B cell, and these IgG will go and bind on the mast cells, and then mast cells will release histamines and it will induce an allergic response from the body. So the mast cells will be histamine production, allergic response from the histamine production. B cells, as we studied, B cells are primary cells which produce antibodies. B cells, right? When B cells encounters any antigen, it divides into different population of clonal B cells, and it mainly divides into a memory B cells, right? A memory B cell which will have a memory, and plasma cells. So this plasma cell is capable of producing large titer of antibodies. So Q1. And NK cells, which are the natural killer cells, they are responsible for ADCC, antibody dependent cell mediated cytotoxicity, right? We discussed the NK cells which are responsible to kill virus infected cells, right? Virus infected cells and tumor cells. Whenever they will not find class 1 MHC molecule, this is very important. Class 1 MHC molecules are always expressed on all nucleated cells, right? On all nucleated cells. This is very important. All nucleated cells will have class 1 MHC molecule. If NK cells don't find any class 1 MHC molecules, they'll just kill off those cells. So antibody dependent cell mediated cytotoxicity, which is third. So the correct Option is C, right? C. Thank you.